You might be familiar with glioblastoma, the deadliest and most common form of brain cancer. However, you might be surprised to learn that it doesn't just attack your brain, it destroys your skull and rewires your immune system. And on top of that, trying to stop the bone loss makes the cancer even worse. Let me break it down for you. The skull marrow is an immune reservoir that supplies immune cells to the brain in response to various injuries. However, the response to glioblastoma was relatively unknown, which intrigued scientists at the Albert Einstein College of Medicine. They studied this by giving mice brain cancer and observing their skulls with micro-CT, which uses X-rays to produce high-resolution 3D images. To their surprise, glioblastoma eroded the calvarium, the region at the top of the skull, particularly in places where the bone fuses together during development. This observation was further mirrored in CT images of human glioblastoma patients, where similar shrinkages were found. Now, why was this happening? Well, to answer this, the scientists genetically engineered mice to express a red fluorescent protein in osteoclasts, our body's bone miners. By tracking these cells through the establishment of the brain cancer, they saw that osteoclast activity was on overdrive in their mice. Through profiling individual cells in the skull and bone marrow, they further found that glioblastoma fundamentally redefines the immune landscape in the skull and bone marrow, leading to more myeloid lineage cells like neutrophils and fewer lymphoid cells like B cells in the skull marrow. Since the bone disruption in glioblastoma was caused by overactive osteoclasts, the scientists want to see the effects of drugs inhibiting these cells like zoledronate, which are commonly used in the treatment of conditions like osteoporosis. Disturbingly, although treatment successfully blocked skull bone resorption, it actually increased tumor aggression and resistance to immunotherapies, namely drugs targeting PD-L1, a protein frequently upregulated by tumors to hide from T cells. It turns out while anti-PD-L1 treatment alone increased activated T cells and further eliminated inflammatory PD-L1 expressing neutrophils as well as tumors, osteoclast inhibitors led to a rebound of inflammatory neutrophils, which could tank more immunotherapy, thus dampening T cell activation. This suggests a potential clinical warning. It might be best to avoid osteoclast inhibitor drugs in patients with glioblastoma, or else we negate the benefits of immunotherapy. In conclusion, brain cancer sucks more than we thought. Thanks for watching and follow for more.